Hi everyone, and welcome to the Chapter 7 lecture for Crowley's An Introduction to Human Disease. This chapter is all about neoplastic disease. So neoplastic, neo is a prefix that means new, and plastic means sort of changing shape. So neoplastic or growth, I guess, is another sort of term for plasto. So neoplastic disease, these are diseases that involve new growths. So these are, all right, we'll get there. All right, so when we talk about cells growing and bodies and tissues growing, all right, we're really talking about the cell cycle. So cells, when they are growing and proliferating and dividing, they go through a cycle where they are sort of doubling their DNA, doubling their parts, growing in size, and also in the nucleus they're doubling their DNA in order to then go through mitosis, which is the process of sort of dividing, that final step of dividing everything equally between two parts and splitting into two cells. So some cells in the body, so during embryogenesis, when a fetus is developing, almost all of the cells are going through the cell cycle. They're doing lots of divisions to grow more parts. But once you are an adult, especially, most of your growth is done. And a lot of these cells in your body, even actually in a newborn, a lot of cells are already kind of in this state, but definitely more so in adults. Once we reach adulthood, a lot of our cells are what we call permanent populations. So these are cells that are no longer undergoing division. They are simply living their lives. They are undergoing all these chemical reactions and they're doing their jobs as cells. In the case of a nerve cell, for example, it is, secreting neurotransmitters, it's doing nerve transmissions, but it's not dividing to make a lot of new nerve cells. There's very few nerve cells that are continuing to populate. All right. And so they're just sort of surviving. They're in a state of senescence, is what we call that. There are also cells in our body that are what we call stable. So they can divide, but very rarely, like once in a while. Maybe if they are injured or something like that. There's a set of cells that are able to divide, but it's not something they're regularly going through. And then there are cells that we call labile cells, and these are cells that are there to continuously divide. And we find these in body tissues that are constantly regenerating. So things like our skin. In your skin, the basement membrane of your skin cells, of your epidermis, are cells that are constantly growing. You form these, they differentiate into keratinocytes, and then the top layer sloughs off. That's exfoliation. So your skin cells are constantly replenishing. There are labile skin cell populations. Also in your gut, lining your intestines and your respiratory tract, the surface cells are constantly kind of turning over. And same thing with your blood. It's why you can donate blood, because you will make more blood cells from your bone marrow. So in the bone marrow, there are a lot of the hematopoietic stem cells. That's a labile population. All right, and it's these labile populations of skin cells, of cells in general, anywhere in your body. These are the cell populations that tend to be the most vulnerable to neoplastic disease, because if a mistake happens anywhere in this cell cycle that causes it to, I guess neoplastic disease, when we talk about neoplastic disease, we're talking about cells that enter this cycle and keep growing and proliferating, not in a controlled way. So there are cells in your body that are designed to sort of be in this cycle, but there's a level of control. The amount of cell division is controlled. Your cells are reproducing as needed. And then there are some populations of cells where they really shouldn't be dividing at all. And so neoplastic disease happens when this cell cycle, when either these cells re-enter the cell cycle and they're not supposed to and they start proliferating out of control, or these labile cells that are supposed to be going through this cycle in a controlled fashion start going through it way too quickly in an uncontrolled fashion. So all neoplastic disease is cell cycling, cell growth, and division in an uncontrolled fashion. Okay, so not all neoplastic disease, though, is cancer, is malignant. So there's really two types of tumors. Whenever you have a neoplasm, you have this uncontrolled growth, you end up with a tumor. And tumors can be either benign or they can be malignant. So benign tumors are sort of just out of control growth. You get too many cells, but the cells themselves are not really abnormal. So in this sort of diagram, cartoon diagram here, you have normal tissue cells in blue, and in red we have the tumor cells. These are cells that have been mutated in some way and now are growing out of control. But they're not terribly atypical, so they look very much like the normal tissue cells. There's just a lot of them. And they also are in sort of a clustered mass that's well defined. There's a well-defined border. And benign tumors can still contribute to disease because they can still, they could press on nerves and cause pain. They can be very large, they can grow very large and just be uncomfortable. So they are not deadly and they do not spread, but they can still be problematic. They still can involve symptoms, and they still can sometimes require treatment. But benign tumors are not the ones that are involved in cancer. So cancer is actually many diseases. There's many types of cancer, and those are diseases that involve malignant tumors. And you can see that malignant tumors are very different in structure than benign tumors. The cells themselves are very mutated, have differentiated quite a bit and no longer look like the normal cells that they were derived from. So they have irregular shapes, variation in their shapes. They have often a larger and darker nucleus than the normal cells. They are oftentimes clustered without a regular defined border, and they can invade other tissues and actually leave the tissue that they originated from. And so there are various stages of malignancy that we'll talk about. But just some examples of some benign tumors versus some malignant tumors. So some classic benign tumors. A lipoma is a tumor of the fat tissue, of subcutaneous tissue, and it can form a sort of bump, a lump, that is often painless but somewhat firm to the touch and maybe unsightly. Sometimes it can press up against nerves or muscle tissue and cause discomfort or pain, and then it can be surgically removed. Hemangiomas, these are common sort of birthmarks, but also can form on adults. Usually we call them cherry hemangiomas, these little tiny little red spots that almost look like red freckles. And these are just outgrowths of blood vessels. And again, they are benign. And uterine polyps and some intestinal polyps. Sometimes polyps are just benign growths. They can also be precancerous, so they do need to be examined, but they can be just benign outgrowths of tissue in organs that have a lumen, that have sort of an inner cavity, if you will. So you can get them in the fallopian tubes, in the gastrointestinal system, etc. Some examples of malignant tumors, carcinomas. These are cancers of the epithelial tissue. So there's lots of different types of carcinomas. So in the skin, for example, you can have a melanoma, you can have a basal cell carcinoma, you can have squamous cell carcinoma. So the different types of carcinomas are named after what cell type they originated in. You can also have cancerous malignant tumors of the connective tissue, so like an osteoma of the bones, a sarcoma of the connective tissue like the muscle tissue. Notice that a lot of these end in oma. Both benign and malignant tumors can end in oma. We tend to associate the suffix oma with cancer, but the suffix oma just means tumor. So I like to point that out because if you're ever in a situation where you get diagnosed with an oma of some kind, you definitely want to ask, is it benign or is it malignant? Don't assume that it's a cancer. All cancers are malignant. All cancers end in oma, with one exception, and those are the leukemias. Leukemia is a cancer of the blood, and emia means condition of blood. Leuco means white. So it's a condition of white blood. It was a historical name because the doctor who first discovered leukemia first described it. He had a patient who had a lot of general symptoms, fatigue, muscle and joint pain. And so he was like, well, let's take a sample of your blood and examine it. And when he took a blood sample from her, it was milky white. He described it as milky. And so he called the condition, whatever she had, white blood, a condition of white blood. And it was because ultimately she had too many white blood cells. It's a cancer of the white blood cells, too many white blood cells. And so it's the one, and it doesn't, because blood is, these blood cells are sort of liquidy. They're fluid, right? Your blood is fluid. The tumors that form are not solid. Blood cancers are not solid tumors. They are sort of fluid tumors. So blood cancers are a little bit unique in their sort of naming, but also in their biology, because it's never a solid tumor that can be removed, like maybe in other tissues that do start as solid tumors. Okay, so just wanted to point out that oma means tumor, not necessarily cancer, and that all cancers will end in oma except for our blood cancers because they're not solid. So how do tumors form? Ultimately, they start with some kind of genetic mutation. So it happens when there's a DNA error that doesn't get fixed in a cell, and it has to be an error that leads to, so it's not just any error, it's an error that leads to misregulation of that cell cycle. So specifically, mutation in genes that cause the cell cycle that promote this cell cycle. All right, and so then that mutated cell now starts to overproduce, to repopulate, or to replicate itself, to divide and make lots more copies of itself. This stage is known as hyperplasia, so an excessive growth, stage of excessive growth, but we're still in sort of a benign stage of tumorigenesis. Then some of those cells, because they're already mutated, and they're now dividing more rapidly, they're more prone to developing additional mutations. So now they start to become even more mutated, and some of those mutant cells are now differentiating, and they no longer are looking like those original cells. They're starting to have more growth potential and also to be different looking. And so they're growing faster, but they're still staying in the same place. Dysplasia is often what is being looked for in a lot of cancer screens or a lot of biopsies. And this is sort of what is referred to a lot of times clinically as precancerous cells. And doctor might want to remove these because of their ability to proceed to cancer. So the early stages of cancer are a large growth with a lot of differentiated cells here that are now a bunch of different cells that are starting to not look like that tissue anymore, like the normal tissue. This is what's called maybe a stage 0 or stage 1 cancer. It's very localized, it's well defined, but it is concerning, and if it's not removed, will continue to grow out of control. It's called an in situ cancer, All right, and this is the earliest stage of cancer. Ideally, we want to catch cancers in this stage so they are very treatable. If the cancer progresses, though, those cells continue to divide and to mutate, and they not only are proliferating, but they also often do a few things to serve themselves. So these cells, in order to survive this tissue, now that there's more tissue here, they need more oxygen. And so cancer cells have an ability to turn on angiogenesis, or to increase the branching of your blood vessels to get more capillaries to feed the tumor more oxygen. And that's a late stage sign, a sign of more advanced cancer. Also, as they progress and continue to divide and mutate, they can also escape the tissue. They can either cause physical damage to the tissue that allows the cells to escape, or they evolve, turn on different cellular mechanisms that allow them to travel. Again, early in embryogenesis, our cells do move, and some cells in our body do move. And so all of the cells in our body have the ability, they have genes that code for being able to travel, but we turn those off typically after embryogenesis. But tumor cells tend to turn on a lot of genes that were on in embryogenesis that allow them to replicate and travel. They sort of revert back to embryonic cells, which is not a good thing for our adult bodies. So they can they increase the blood vessels, the blood vasculature to the tumor, and then the cells can invade the vasculature through that those new blood vessels. And if the tumor cells get into the blood, they can use the blood vessels to travel to other sites and invade other sites. And that traveling of cancer cells is called metastasis. Metastasis is the term for cancer moving to distant sites in the body. So if you have a cancer, let's say someone has breast cancer, all right, the primary site, those cells are breast cells that have mutated and started to proliferate out of control. And if the cancer progresses to those later stages, to metastasis, and those cancer cells end up in the blood, we call those circulating tumor cells, or sometimes you see them as CTCs. All right, then they can leave the blood vessels and enter other tissues and form tumors in those sites. And so someone who started with breast cancer may also then have cancer in their liver or their lungs or their brain, but it's not brain cancer, it's not cancer of that started in the brain with brain cells growing out. It's a secondary site. It's a metastasis of the original breast cancer. Sometimes people are first diagnosed with cancer at a later stage, and they maybe are diagnosed through a radiology screen like an MRI or a PET scan. And if they have multiple spots where there's cancer, you can't necessarily tell from that PET scan what type of cancer it is. You have to do a biopsy. You have to test the cells to see what tissue they originated from. Are they breast cells? Are they liver cells? Are they lung cells? Where did the cancer start? And so those biopsies, testing the cells to see what type of cells they originally were, is important for being able to treat the primary site where all the cancer cells are originating from. So let's get back to the sort of the cellular basis of cancer. What happens when we have these neoplasms, these new growths? So normally cells divide, they make a copy of themselves, and then you end up with two daughter cells, all right, and then they divide again, but maybe one of the daughter cells ends up with a DNA mutation, okay? Generally, if a cell is mutated, if the DNA is mutated, there's a couple of things that the cell can do. It can either fix the mutation. There are ways that we have DNA repair mechanisms to fix mutations. Or the cell will apoptose. it'll go through apoptosis. It'll basically say, I'm fooked, and so I just need to end this lineage and self-destruct. All right, that apoptosis is that programmed self-destruction that cells have when they are misformed. All right, but sometimes cells with certain DNA mutations continue to divide and grow. And mutations tend to beget more mutations. So the mutations that don't result in apoptosis tend to be mutations in this cell cycle. So the cell cycle, and I'm not really going to go through all the specific stages, but it involves growing, doubling, one's proteins, the size of the cell, also doubling the DNA in the cell in order to get ready for mitosis, which is the actual splitting, dividing of the cells. And along this cell cycle, there are multiple checkpoints that the cell has, where literally these are quality control checkpoints to check, do we have the right number of chromosomes? Did we replicate the DNA properly? Are there any breaks or mutations in the DNA? Is everything in order before we get to this point in mitosis where we're going to divide the cells? So there are multiple mechanisms the cell has, these quality control points where the cell stops and says, is everything okay? If it's not, it's supposed to undergo apoptosis, but sometimes it doesn't. And these control points are skipped over, and that's when we start to get uncontrolled cell division, which can then lead to additional mutations. So one mutation is not going to lead to cancer. What cancer and truly neoplastic disease really involves multiple mutations, at least 4, according to most textbooks, but some cancers more than others. And so it's really an accumulation of these mutations. They mutate, 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 and then once an accumulation of mutations has been achieved, then they really become out of control. So these are cells that are dividing unchecked and are able to undergo more mutations until a point where they just are dividing out of control. And that's where we get neoplasms, where we get cancer. So what are things that can cause these mutations? Well, there's lots of things that can cause mutations, which are DNA, at the DNA level. These are genetic changes that occur. So lots of different things in our environment that our cells are exposed to can lead to mutations. For example, radiation. Ultraviolet radiation from the sun is not very penetrating, so it doesn't cause mutations deep in our tissues, but it can cause mutations of the skin, and we are covered in skin. So our skin regularly exposed to ultraviolet radiation, that can lead to cancer. It can lead to DNA, genetic changes in the skin cells that lead to uncontrolled proliferation. Also, other types of radiation like X-rays, exposure to X-rays or exposure to radioactive materials. If you work in a field where there's, radioactive materials, those can penetrate more deeply and cause mutations of more core tissues, including the bone marrow. Like we said, the skin has a lot of labile populations of skin cells that are regenerating. So does your bone marrow. So bone marrow tends to be very sensitive to radiation, DNA damage. Infection is another source. There are various viruses and even bacteria that can cause genetic mutations, but the most well-known ones are HPV, human papilloma virus, and the hepatitis viruses, namely hepatitis B and hepatitis C. These are viruses that are known to cause genetic mutations in the cells they infect that lead to the development of cancers of those tissues. P.S. There is a vaccine for HPV, human papillomavirus, and also for HBV, hepatitis B virus, but there is no vaccine for hepatitis C. So that one is still a big concern. Various chemicals in our environment that we're exposed to, whether they are things that we consume in our diet or things that we breathe in, like cigarette smoke contains various chemicals that can lead to cancer. We call those carcinogens. So it's important to avoid carcinogens to try to prevent those mutations in the DNA of some of our cells. Another thing is just the natural process of aging. As we get older, our cells continue to divide and replicate, and every time our cells replicate, there is a sort of baseline error rate of our DNA polymerase that copies our DNA. All right, just like when you type a paper, you're bound to have some typos. And yay for spell check and grammar check that helps us correct some of those mistakes. Our cells also have DNA repair mechanisms, but the longer the paper is, the more likely you're going to have a typo that doesn't get caught, right? And so same thing, the longer you're alive, the more times your cells divide and replicate, the more likely it is that you're going to end up with an error that went unchecked. And so it's just the natural rate of error times time leads to DNA damage. And then another one that I added here is chronic inflammation. So we talked about this when we talked about inflammation, that all those cytokines that can damage tissue, they can also lead to DNA damage if you're exposed to them long term. So inflammation is good in the short term. It does promote healing and it does help to fight off pathogens. But chronically, long term, it can lead to damage that can sometimes be in the form of DNA damage, which can then lead to cancer. There's a lot of chronic inflammatory diseases like gastrointestinal reflux disorder or gastroesophageal reflux disorder. Constant inflammation of the esophagus is a risk factor for esophageal cancer. So all of these things though, all of what they all have in common is that they can lead to damage of the cell's DNA, and that's DNA damage can be, if it's in a gene that has to do with the cell cycle, can lead to uncontrolled proliferation. So which genes are likely to contribute to cancer? It's going to be genes that are involved in this cell cycle regulation. Cells growing and copying their DNA and then ultimately splitting into two daughter cells and dividing that DNA equally. All right, there are these checkpoints that the cell has, but they can be overridden. All right, so the types of genes, mutations in genes in these four categories are the ones that are likely to lead to cancer as opposed to sickle cell anemia or achondroplasia or any other disease of a body system. So I've given them sort of icons. So we call these, any gene that a mutation in it can give rise to cancer is called an oncogene. Onco means cancer. You go to an oncologist if you have cancer. Not to be confused with the suffix oma, which means tumor, which not necessarily cancerous. Onco is cancer, oma is often cancer, but not necessarily. All right, so there are proto-oncogenes. These are genes that are usually growth factors of some kind. They are genes that make this cell cycle go. I like to think of them as sort of like the gas pedal, right? And if your gas pedal is broken and it's like there's a brick laying on your gas pedal and you can't get the brick off, all right, you can't slow down your car. You want to be able to go fast sometimes, but you want to be able to control the speed of your car, right? Just like cells need to be able to control their cell cycle, when they stop and when they go, when they divide and when they grow. And so if there is a gain of function mutation, so if they're making too much of these go genes, of these genes that make the cell cycle go, that's not good. Another class of genes are the tumor suppressor genes. Now, these are genes that are like the brakes on the cell cycle. All right, normally, and again, with this driving analogy, you want your brakes to work because sometimes you need to be able to stop your car, just like you need the brakes on the cell cycle to work so that it can stop the cell from growing and dividing if it's mutated. All right, but if these tumor suppressor genes are broken, then the cell cycle loses its brakes. It loses its ability to stop, and so it keeps going. Another type, sort of category here, are the DNA repair genes. So if these cancers are due to DNA mutations, we can prevent cancer by fixing those mutations. Our cells have tools to repair DNA breaks and DNA mutations, but if those tools are not there, if those tools are knocked out, all right, then they become more vulnerable to these mutations, and that can increase the risk of cancer. And then lastly, we know that if the cell cycle is out of control, if there's some damage or mutation, that cells are supposed to undergo apoptosis. They're supposed to self-destruct. But if that self-destruct feature is turned off, then they keep growing. So these are the 4 categories of genes. Genes in these 4 categories commonly lead to cancers. It's mutations in these categories specifically. So some common proto-oncogene examples. These are ones that are basically growth factors, and these are gain of function mutations that, so when they just start to be overproduced essentially, which increases cell division. So 2 of these that you may have heard of, EGFR, which stands for like epithelial growth factor receptor, I think, and HER2. HER2 is commonly characteristic of breast cancers. So a lot of times breast cancers will be, oops, ignore that, will be looked at and to see what type of mutations the cancer cells have. So you may hear a type of cancer described as HER2 positive or not, meaning that, and that can help dictate how a cancer is treated. Depending on which types of cells in the cancer are mutated, determines which types of therapies are going to work best. All right, so these are some examples of some and these are receptors in the cell surface that signal, that trigger basically a chain reaction that leads to cell division and DNA replication. It tells the cell it's time to grow. So if there's too many of these, it's telling the cell too much to grow. So these increase cell division, which is a pro-cancer kind of thing. A common example, a classic example of a tumor suppressor gene that's often mutated in cancer cells is this guy here, P53. This is the protein P53, and it's encoded by a gene that's called TP53. And it's a very important central regulator of the cell cycle, of apoptosis, of DNA repair. So it's kind of like does all the things. And it's really one of the important checkers in that, at those checkpoints, right, in cell cycle regulation. P53 plays a big role in kind of checking whether the cell has any of these types of damage, and then directs whether or not cell division happens or whether the cell is going to apoptose. And there is actually a sort of heritable cancer condition that's known as Li-Fraumeni syndrome, and these are people that have inherited a 1 mutant P53 gene. And so if they already have 1 gene that's mutated there, they only need to undergo 1 other mutation, critical mutation, in order for cancer to develop. And so they basically start off with all of their cells being predisposed to developing cancer. So they have to be very careful and undergo a lot of screening to look for cancer because they're just prone to developing cancer. Not any specific type of cancer, but any, like, of all of their body tissues. So breast cancer, colon cancer, whatever, and all tissues in their body already have this one mutation, and so they only need a couple more mutations in order to develop cancer. So there's a very high rate of cancer in people with this particular genetic mutation. Another gene that contributes to increased risk of cancer is the BRCA gene, BRCA. There's BRCA1 and BRCA2. And these are, the BRCA gene codes for a protein that does a lot of stuff, but one of the things it does is it aids in DNA repair. And so people who have these mutations are less able to repair damaged DNA. And so that, again, they are more prone to certain types of cancer, particularly breast cancer. So this gene's particularly active in breast tissue. And so this is just showing you the relative risk of developing breast cancer if you have a mutant BRCA1, a mutant BRCA2 versus a normal BRCA BRCA gene. So the risk is much higher if you have mutated BRCA genes, because you have less ability to repair DNA damage in that tissue. So this is a loss of function mutation. These are mutations that cause the BRCA gene to lose its function of being able to repair genes. So some of these are gain of function, like with the proto-oncogenes we're looking at. These are genes that gain a function. They are overexpressed and overzealous. They make the cell cycle go too much. Versus tumor suppressor genes and DNA repair genes, these are things we need to prevent cancer. And so if we lose these are loss of function mutations where we lose the functionality that we need. And then lastly, that last class, those apoptosis genes. This is one where it's another gain of function, and it's a little bit confusing here, but BCL2 is an important protein in the cells that inhibits apoptosis. It stops the cell from doing apoptosis. Which we need because, we don't want cells just self-destructing all the time. We only want them to self-destruct when they're supposed to, all right? But if we have too much BCL2 in our cells, all right, if there's a gain of function mutation and the BCL2 is overactive, then it will inhibit apoptosis of cells that do need to self-destruct. And so overexpression of BCL2, a mutation that makes for an overactive BCL2 protein, inhibits that needed apoptosis of cancer cells and can lead to cancer as well. So those are just some different categories of genes that contribute to the development of cancer. So another thing that contributes to cancer is the immune system. So we talk about the immune system mostly in terms of preventing infection and fighting infection and foreign substances that invade the body, all right? But cancer is also cancer is weird because it is self, right? It's our own tissue that's proliferating, but it's abnormal tissue. And so we do have specialized immune cells that are trained to recognize and destroy cancer cells. Most commonly, these orange guys here are the NK, the natural killer cells, and that is sort of the primary class of cells that hunt and kill cancer cells. And these are white blood cells of the immune system. Another important cell type are those cytotoxic T cells, the CD8 positive killer T cells. All right, these guys can also recognize certain cancer antigens and kill cells. So when the immune system is working properly and identifying cancer cells by their atypical receptors that they have, maybe, or turning on some of those characteristic embryonic traits that they're not supposed to have. All right, then the immune cells can recognize them and eliminate them. Probably everyone watching this video has had cancer, has developed cancerous cells in their body, but their immune system has taken care of it and we didn't never realize it. But cancer cells are constantly replicating and mutating and evolving and adapting to their environment. And sometimes they are able to actually escape recognition by the immune cells. And so they do this in a number of ways. They can change the surface markers that they have on their surface, so they no longer, the immune cells don't recognize those antigens anymore. They also can secrete cytokines, secrete chemicals that suppress immune cells. And also sometimes the immune system just gets exhausted. The T cells populations can just become overexhausted, and also the cancer cells can grow very densely, such that the immune cells just can't physically infiltrate the tumor site. So there's a lot of ways in which cancerous tumors actually escape the immune system, evade the immune system. So that also leads to disease. So what are some of the signs and symptoms of cancers? I want to stress that cancer is not a single disease. It's really cancers. I used to work, I did a research stint after college at the National Cancer Institute, but I think I don't know if they have renamed it or not. I should check into that, but it's really should be the National Cancers Institute. This is just 3 different sort of signs that have symptoms of different types of cancer: blood cancer, breast cancer, colon cancer. Okay, there's no universal sign of cancer. Cancer is hundreds of different diseases, and it depends on where that cancer originated, what type of cells have become cancerous, have started to overgrow and proliferate. And so it's important to know the different signs for different cancers. And a lot of times they involve changes to that system. So for example, breast cancer, it basically noticing anything atypical in your body functioning can be a sign of a disease of that system. And cancer is always on the differential, the diagnosis differential. So these signs of colon cancer can be signs of any number of diseases of the gastrointestinal system, but cancer is always one that you would want to explore and rule out for sure. So how do we diagnose cancer? Well, there's a lot of different ways that we can test. So the first and most important, I would say, is screening. So screening tests these are like annual or every 5 years, every 2 years, a test that you undergo to look for cancer, not dependent on symptoms at all. So this is pre-symptomatic. So screening is a prophylactic test. This is preventative, trying to catch things early before you even have symptoms. So it's a good way to catch those early stage cancers when they're still very treatable. So things like mammograms, which is what's pictured here, to screen for breast cancer. Pap smears, these are samples of cells taken from the cervix in the vagina to check for cervical cancer, and those are done oftentimes annually for women, to check for cervical cancer caused by HPV, human papillomavirus. Another one is colonoscopies after, I think, age 50 or younger. If cancer runs in your family, people are recommended to go through regular colon screening, colonoscopy, a procedure to look for colon cancer, essentially. If something is found on one of those screens, so like if there's a lump in the breast tissue or a polyp in the intestines, the doctor may want to do a biopsy, so take a sample of those cells, those abnormal cells, and look at them under a microscope. So that would be cytology or cytological or histological testing, looking at them to see are there signs of dysplasia, do the cells look normal for that tissue or do they look distorted? And so that they can kind of stage what that is, the tumor benign or is it malignant? And if it's malignant, what stage of malignancy? There are some cancers, but very few, that can be tested with a urinalysis, but it's on here, so I'll address it. Some hormones are produced, some certain proteins are produced and can be excreted in the urine. Two that I can think of off the top of my head: PSA, which is the prostate antigen, prostate serum antigen maybe, is something that is produced by prostates when they have tumors and is a non-invasive test, a way to screen for prostate cancer. Also, sometimes testicular cancer can cause the cells of the testes to start producing HCG, human chorionic gonadotropin, which is the classic pregnancy hormone. So sometimes I've heard lots of stories on the internet, like on Reddit, where somebody, a guy took a pregnancy test as a joke and it tested positive. That is a potential sign of cancer, of testicular cancer. So definitely was encouraged to go to the doctor. Blood tests, so all those same chemicals, and also sometimes, remember, if a cancer has metastasized, those cancer cells themselves might be detected in the blood if it's entered the circulation. So blood tests, urinalysis tests, these are not definitive tests, but they might be clues that indicate further diagnostic tests need to be done. A lot of times imaging tests are very telling. So MRIs, X-rays, and PET scans can all help to detect tumors. A mammography is a type of X-ray essentially. So it's a type of radiology exam. Ultrasounds also a type of imaging that can be very helpful in identifying cancers, I should say. So what are some of the treatments for cancers? There's many, and every cancer really is a different disease. And by that I mean, like, breast cancer is not even a single disease because there are lots of different types of breast cancer depending on what genetic mutations have accumulated in the cell. So cancer is just very vast category of diseases. The thing they all have in common is that the cells are proliferating out of control. The cause of that out of control proliferation can be different for every single cancer due to different gene mutations in different tissues and different cells. And so cancer treatments oftentimes have to be very personalized. Some of the different options. I'm going to start with surgery. All right, so for a solid tumor in a tissue, sometimes it can be, especially if it's a stage 1 or 2 tumor, it's local, it hasn't spread to other tissues. The cancer can oftentimes be treated simply by being cut out and removed. It has to be done very carefully. If like a single cancer cell is left behind, it could continue to proliferate and spread. And so it has to be a very well-defined cancer in order to be able to be treated just with surgery. Sometimes it will be treated with surgery and then also with additional therapy like immunotherapy, radiation therapy, or chemotherapy. I'll get to chemotherapy and immunotherapy in a minute. Radiation therapy is using X-rays. So X-rays we know can sort of cause cancer because they can damage DNA, but if we use them in a targeted way, we can damage the DNA enough to kill the cells, and that's kind of the idea. So radiation can be aimed at sort of specific tissues. You can place little radioactive chips into a tumor to irradiate just the local tissue. So it's a little bit more localized. It kind of can target the general tissue area of the cancer. Bone marrow transplants can be used to treat blood cancers. So basically blood cancers, you can give someone radiation or chemotherapy to kill all of their white blood cells, the cancerous ones and the non-cancerous ones, and then replace their bone marrow, replace the diseased bone marrow with healthy bone marrow as a form of treatment for cancer. Bone marrow transplants are risky procedures though, and it's risky to ablate all of your immune system for a brief period of time, and transplants are not always perfect. Sometimes transplants don't take. You have to have a really good match, and so it's not a cure-all. Not everyone can find a transplant and have to go through these other therapies. Hormone therapy can work for certain types of cancer cells that are triggered by hormones or inhibited by hormones. So again, not every cancer is going to need or respond to hormone therapy. So now let's talk about chemotherapy. Cancer chemotherapy is generally not very specific or targeted. So there's no drug that magically is specifically toxic to cancer cells. Generally, cancer chemotherapeutic agents, cancer drugs, they tend to target cells that undergo replication. They tend to target cells and be toxic to cells that are labile, that are constantly undergoing cell division. And so the cancer cells are not the only cells that are labile populations in your body that are fast replicating. And so a lot of the common side effects of chemotherapy are due to the fact that the chemotherapy drugs also target healthy cells that are undergoing a lot of replication, including cells in your hair follicles, which is why hair loss is very common as a chemotherapy side effect. Fatigue and anemia and bruising all have to do from the fact that your blood cells constantly replicate. So that fatigue is usually an effect of the anemia because you have less ability to transport oxygen because you're killing a lot of your blood cells. It also makes you prone to infection because of killing off a lot of white blood cells. And nausea and diarrhea and vomiting are also a common side effect, and that's because another population of labile cells in your body is your gut lining. Your gut cells constantly regenerate similar to your skin cells, and so you're just kind of killing off massive amounts of gut cells, which leads to gastrointestinal kind of symptoms. And so chemotherapy drugs are not ideal because really you're poisoning a lot of the cells in your body in hopes of killing the cancer cells and recovering, and those other cells recovering. But it's a very, very sort of toxic treatment and not pleasant, but oftentimes very successful because usually no one replicates as fast as those cancer cells do, so they're particularly vulnerable to those chemicals. An area of heavy research and development and sort of that's revolutionizing a lot of cancer treatment are various immunotherapies. So to go back to this slide here, immunotherapies can include using cytokines or cytokine blockers. That's part of the immune system. And so a lot of these immunotherapies are basically just trying to boost your own immune system's ability to kill cancer cells, because remember, they can do that. So trying to help the immune system to kill the cancer cells. So we could give cytokines or cytokine blockers. There are some immunotherapies that involve antibodies, so giving people monoclonal antibodies that recognize the cancer cell and help the immune system to recognize and target So we can do antibodies. I'm going to say immunoglobulins, IG, immunoglobulins. And then the third type are cell therapies. And these cell therapies have been very successful, and that's what I want to talk about. So the two types of cell immunotherapies are TIL, which stands for tumor infiltrating lymphocytes, and CAR-T therapies, which stands for chimeric antigen receptor T cells. So they're similar but different. So let's talk about the TIL therapy. So in this therapy, you take the tumor out of the patient and you dissect the tumor. The doctor would do this, and they would pull out all of the white blood cells that are in the tumor. So all the white blood cells that are already fighting this tumor, that already recognize it. And then in the lab, they would kind of screen those white blood cells and test them to see which ones are the best at recognizing and killing tumor cells. And then they would select those and grow up a lot more of them and then transfuse them back into the patient. So basically, you're taking white blood cells from the patient, specifically white blood cells that are really good at killing the tumor, and you're expanding them. You're growing up more of them in order to feed them back into the patient. And these work well, but it's time consuming, it's labor intensive, it's expensive, and has to be done for each patient. So you can't sort of scale this up. This is patient specific and very tailored, but it can be very successful. Another type of therapy is taking just taking blood from the patient, just taking their blood and isolating some T cells from their blood, just any old T cell, and then taking that T cell to a lab where it gets genetically modified with a T cell receptor that has been determined to work really well against that type of cancer. And so you're genetically modifying their T cells to be really good killers. And then you grow up more of those genetically modified cells and you infuse them back into the patient. So this is a therapy that might be more scalable. It does require you to take the T cells out of a patient, but just a blood sample. You don't have to actually take out the tumor and do a surgery. And you don't have to go through this labor-intensive process of screening their white blood cells to regrow them. You just take any old any old T cell and genetically modify it. And those genetic modifications could be somewhat generalized. There could be some marker on 70% of colon cancer cells that works really well. And so this could be a process that is more automatable and more scalable for more people. So this has been a really promising therapy. They're very similar, but this one uses genetic modification and technology to make really good killer T cells, and this one is just kind of trying to select the naturally good T cells in a patient already. Both processes are using the patient's T cells and feeding it back to them after modifying them in some way. So that is where I'm going to stop at the end of chapter 7. Thanks for watching.